we developed this reporter system by genome engineering colon cancer cell lines to have a fluorescent readout, a visual readout of the stem cell side and the maturation side. That's where we want the cancer cells to move towards so that they properly mature and eventually die as part of the natural life cycle of the colon. In order to sort of develop what we would think about as a screening tool for drugs, we want the drug to shut off the stem cell side so that signal should go away and the signal on the maturation side should be turned on. So that's the platform we set up. And then we screened drugs and we identified an epigenetic regulator that's being inhibited by a small molecule turned on the differentiation reporter to the strongest extent in this pool of drugs. So we got excited about that finding and we wanted to understand how this drug is working, why is it promoting differentiation in colon cancer, and really it's coming back to that original point about the shape-shifting property. We want to limit that and not allow the cells to stay in the stem-like state. And I think that's going to have therapeutic value, and it's a different approach than traditional therapeutics. What I think the strongest takeaway is the concept that if we push the cancer cells towards maturation and reduce this plasticity that cancer co-opts in the GI tissue and in particular colon tissue, that is a strategy that we need to keep hitting on. Different approaches, different drugs, different ideas, different models. If this work, I mean, I hope it really helps in terms of moving HDAC1/2 inhibitors in a meaningful way towards the clinic. But if not, if there's just too much toxicity or it's prohibitive in other ways, I hope the concept really lives on and we can really, I think that's, deep down, I feel like that's where we're going to find a therapeutic benefit for patients.